Thanks for the introduction. So today, I will show the topic about a candle drug. We just do some experiment right now just on the animal models, which is for the aspirin. You guys all know this drug, right? But we just combine with another Chinese compound with called curcumin, and to combine it together and to produce the Aspirin Curcumin Ester. Also, those drugs, we need to make it go through the blood-brain barrier, so we need to load it with a Biomimetic Nanodrug, and to show this drug can improve cognitive deficit in the Alzheimer disease models. Right now, it's only. So the topic today for me, is for the drug. How do this for the Alzheimer disease? At the beginning, let's introduce some background for the Alzheimer's disease. So this kind of disease, as you guys know, is a kind of neurodegenerative disease. Right now, almost 35 million dementia patients worldwide. I think they have more. And the most common types of the dementia lead to the cognitive impairment. And right now, it's the third largest death fact. And also, the pathogenesis is unclear, and in clinic, they have no effective treatment has been found. So for the research field, right now, this kind of disease has become the most popular international topic. And recently, lots of the paper have been published in Nature Science or Cell, and all of them are related with the target or some drug or mechanism of this kind of disease. So for this disease, we just know the pathologic hallmarks. It just gets the extracellular beta amyloid and the intraneural, the microtubule associated protein tau gets a high phosphorus. And also, they have a lot of neuron loss. So they have just get the brain atrophy. So right now, they have a lot of hypotheses. And also, they have two kinds of the major drug in clinic to treat this kind of disease, but still cannot treat it, even just improve some symptoms of this kind of disease. So for the cholinergic hypothesis, it just can, this kind of drug approved by FDA for clinical use can only relieve some symptoms, and it is difficult to fundamentally reverse the development of this disease. Also, for the beta toxicity hypothesis and for the tau hyperphosphorylation hypothesis, right now is a development target for these two targets. Only right now with high hope are or failure. So as we know, this kind of disease is a mute factor of the complex disease. They have lots of the pathogenic hypothesis and main susceptible gene of the AD, and also this susceptibility gene are involved in the pathogenic of the AD. So current situation and bottleneck of the drug research, right now they have lots of drug in different phases of clinical trial, but until now, they have no good news for this kind of drug. So we need to think about how this kind of drug are failed. Right now, we just focus on this complex disease for the single target or single drug. So we just compare, we just know that this kind of disease is quite complex. So we need to know they definitely have the multi-target AD. In Chinese medicine, they have a natural medicine, also have some mutual compounds can get the advantage of the mutual target of the Chinese medicine. So we can combine action of the active substance to meet with the target of the mutant target of this candidate. But these compounds are complex and the mechanism also is elusive. So the target and the mechanism of the effective substance are also unclear. There also is kind of our Chinese medicine bottleneck. So right now we know the aging is a major reason of this kind of disease. So also we noticed for the metabolic syndrome also is another major reason for this kind of disease. So they have several evidence to show this kind of disease we just mentioned for the Alzheimer disease are related with some metabolic syndrome, such as for the diabetes and for the obesity, or even we use high fat to induce some insulin resistance can promote the AD transgenic mice get more the amyloid or inflammatory activation. So some people even say for this kind of disease cause is for the type 3 diabetes. So we just want to mention for these two major reasons for this kind of disease, whether we need to focus on these two parts. So first we use some animal model to get the high fat dent can induce the enhanced inflammatory response in the animal model, which is used for animal model for APPS1 animal model. And also, we combine with this model with another diabetes mice model, we call it MKR mice. We also can see the inflammatory response is increased in the brain, even in the hippocampus or the cerebrum. So based on this reason, we just use one kind of Chinese compound we mentioned for the curcumin, and get combined with aspirin. We know that is COX enzyme inhibit can reduce the inflammation. And for the curcumin, actually this one can adjust some metabolic syndrome issues. So we just want to combine these two drugs together, use some the biosynthetic process we call is for the aspirin curcumin ester. And then at the beginning, we show since the aging is a major issue for this Alzheimer disease, so we use the C. elegans to test whether this drug can increase the lifespan. And our results show definitely these compounds definitely can increase the lifespan and also can get increase for the lipofuscin and the reproduction of the C. elegans. Also, we tested the mechanism of the CA. Since I don't know whether you guys can do some experiment for the C. elegans, but I will explain to you for the SK1. It's just like the one compound along with the insulin pathway, signal pathway. So our results show for the CA, we just mentioned, is for the curcumin aspirin ester, extending lifespan is related to the regulation of this SK1 transcription factor with the antioxidant. So just get related with DEF2, ET2, or SK1 mutation and the oxidations, and also show the CA can extend. And also, this drug can promote the entry of the SK1 into the nucleus and can activate the downstream of the antioxidant gene, for example, for the GST4 and the SOD3, to reduce the production of the ROS. So, based on this result, we just try to do another experiment for the mice at this level. So we just, at the beginning, we use directly for the CA. Cannot see good effect, since, as you know, for the curcumin, it's not a good one to have a good biology process in vivo. So we try to deal with this problem. We just use the synthesis process with the nanocomplex to prevent the biology for the process. For example, we just use mimic. We also call this for the hybrid membrane. So this hybrid membrane, we just combine with the red blood cell membrane, combine with the microglia membrane, and the ratio is 1:1. Why we choose the RBC membrane? Just want to prevent this drug degradation in the blood. And then we use glyphosate oxide combined with PEG200, which is kind of nano complex, and combined with the CA, and to test whether this nano complex can prevent the animal model, which is Alzheimer's disease animal model, whether how can improve the learning or memory, or whether can just for ameliorate for some pathological result. So first, we need to test for some just for the characterization of this nanometallize. So we use trans 11 microchrome for these nano particles, and also we use the zeta potassium analyze for both of them and get the interpret efficiency and the drug loading capacity of the nanoparticles. And in vitro, we also test the drug release and uptake of these nanoparticles. So for the cumulative release of the CA from the GP or the combined together as a pH 7.4, and also get the imaging for the fluorescence intensity analysis of the cell uptake and also get the image of this cellular localization of the BV2 cells, which is kind of the microglial cells of mice, and also test the cell uptake for this in presence of the different inhibit. And also need to test the biocompatibility of this nanoparticle. So first we need to test whether they have some cytotoxicity, and also they get some quantification analysis of the hemolysis assay of different nanoparticles. And also test some quantitative for the quantitative analysis of the raw cells, which is kind of the microglial cells. So we also need to test for the PK for pharmacokinetics and also for the biodistribution. We use animal imaging to test different time for 4 hours, 8 hours, and 12 hours for the animal, the drug to whether into the brain over, and also test the biology distribution in various organs, including the heart, liver, spleen, lung, and the kidney, and the brain after 12 hours. And also need to quantitative analyze of the fluorescence signal intensity. Those pictures just for the representative fluorescence image of the blood sample for that. So based on this test, then we just injection into the mice and to test whether they have some function for learning skill and memory of the animal model. So we just did the water maze and show definitely can improve the learning and the memory skill. And also we show this drug can improve the learning skill for next. And also this drug can show the neuroprotection according to the spin density and for some the synapse related protein, and also the synapse test for emergency. And also we show this nanoparticle of this drug can reduce the beta production or microglial activation regulation, especially for the M2, decrease the M1 type of the microglia and increase the M2 level of the microglia. And also, we test the in vivo toxicity of this nanoparticle of in vivo. So for this result, we just get summarized. And this kind of drug for the CA can increase the lifespan of the C. elegans with or without the oxidative or heat shock stress. Mostly through the upregulation of the low scammage enzyme, including SOD3 or GST4, and for the DEF16 transcription activity may medic CA effect. Those DEF16 is related with the insulin signal pathway. And also, this kind of drug loaded with nanocomplex with anti-inflammation and neuroprotection function was developed for Alzheimer's disease by polarizing microglia. And based on the higher brain target ability for the bioavailability and for the biocompatibility. And the biomimetic nanocomplex could be a powerful tool to deliver drugs. This time we just test for the CA, and also can use this for the nanocomplex to deliver another drugs against the Alzheimer's disease. Since actually for the we just mentioned for this mix of the nano particle, they also, even though they don't have the drug, they also have some anti-oxidative effect. So for this drug, I just want to thank for my labs, the PhD and for master, and also thanks for the collaboration of the Professor Tu Haijun, which is in Hunan University, and for the University of Calgary of Professor Zheng Xilong. So we thanks for the funding support for the national science funding of Chinese. Okay, thank you for attention.